the cell is? Yes, Mr.
Yes. A cell is a basic unit of life.
Yes, a cell is a basic unit. Okay, Martin?
A cell has various components such as a cytoplasm, nuclear membrane, and nucleus.
And a nucleus. Thank you for mentioning the nucleus because our main focus will be on a nucleus today. So, as Umahle said that a cell consists of various components, including a nucleus. So, a nucleus, this is the structure of a nucleus. This is where mitosis takes place. So, what do we mean by mitosis? Mitosis is a biological process whereby a single cell divides to form two identical daughter cells. So, before we delve deeper into mitosis, I want you to familiarize yourself with the keyword chromosome. So, a chromosome is a thread-like structure that is made up of DNA and proteins that carries genetic information. Okay, so now let me show you the structure of a chromosome. So a chromosome is made up of two chromatids. This is a chromatid, right? Oh, so here is the other chromatid. Yeah, this is also a chromatid. And then we have a part here that connects the two chromatids of a chromosome. So this part is called a centromere. Centromere. So this whole diagram is called a chromosome.
Yes.
Since I've already showed you how the structure of a chromosome, now let us go back to our main topic, which is mitosis. So mitosis is the process by which a single cell divides to produce two identical daughter cells, each with the same number of chromosomes as the original cell. So let us say this is our cell, right? So if our cell divides and forms two identical daughter cells like this, so as the cells in two that are produced by this original cell will share the same number of chromosomes as this original cell. Do you guys understand?
Yes.
So mitosis allows our bodies to grow, repairs tissues, and replaces old or damaged cells. Take for example when you get a cut or how your body grows from a baby to an adult. So what happens is that mitosis generates new cells. So mitosis occurs in somatic cells. What do we mean by somatic? Somatic cells are body cells which have a nucleus that contains the cell's genetic material. Mitosis consists of 4 stages. We can use PMET to know the sequence of the stages. But before mitosis occurs, we have a stage which is called interphase. Interphase is where DNA replication takes place. It is also where cell growth takes place. Organelles are duplicated in this stage. And lastly, this is the stage where preparation for cell division occurs. And at the end of mitosis, there's also a stage which is called cytokinesis. Cytokinesis is the final stage of the cell cycle. Now moving on to our stages of mitosis, we have P that stands for prophase, M for metaphase, A anaphase, and T as telophase. So let's start with prophase. Before we delve into what is happening in prophase, let us first look at the structure of the cell. So a cell consists of a plasma membrane. A plasma membrane is responsible for controlling what goes in and out of the cell. And then we have a nuclear envelope. Nuclear envelope is responsible for what goes in and outside the nucleus. So this is a cell, and then this is a nucleus. And then we have a chromosome. A chromosome two chromatids, and these two chromatids are joined together by a centromere. And then we also have a nucleolus and a cytosol, which is a fluid that is found in the cytoplasm of a cell. Moving on to the centrioles. Centrioles are responsible for the formation of spindle fibers. And then, as you can see, spindle fibers, they are thread-like structures. So these spindle fibers are responsible for chromosome movement. Do you understand?
Yes.
Okay, so now let us move to what happens to prophase. So what happens is that the double-stranded chromosomes condense and become visible. So when we say they condense, we mean that they shorten and become thick, right? So in the first stage of cell cycle, which is interphase, this is how our cell looks like. Okay.
Am I wrong if I say spindle fibers are spindle threads?
No, you are correct. You can call them spindle fibers or spindle threads. It's up to you. The one that you're comfortable with, you can use it. For example, if you take a look at the interphase, or the structure of the cell.
I can't see clearly on the bottom. Can you please write on top of the bottom?
Okay, it's fine. Okay, let us look at the structure of the cell during interphase. So our cell looks like this. This is our nucleolus, and then we have this chromatin network. So you see the difference between interphase and prophase, right?
Yes.
So during interphase, we have chromatin network, and then here at prophase, we have visible chromosomes, right? It is because those chromatin networks, they condense to form visible chromosomes. Each chromosome consists of two identical chromatids. If you take a look at this chromosome, you can see that this chromosome is identical to this one, and also this one is identical to this one. If they were not identical, you're going to see a chromatid that is purple and the other one that is red. So you can't see something like that. They need to be identical to one another, and they are joined to each other by what is called a centromere. So moving forward, the nuclear membrane, which is also known as the nuclear envelope, and the nucleolus disappear. And then we have spindle fibers that are formed by centrioles. Spindle fibers form as centrosomes move to opposite poles of the cell. Centrosomes is centrioles, guys, where they begin creating a network of fibers called the mitotic spindle, which will later attach to the chromosome and help pull them apart at a later stage, which is called anaphase, but we will delve deeper once we figure out anaphase. Moving on to metaphase. Metaphase, it is whereby chromosomes align and arrange themselves in a single row in the center of the cell, which is also known as the equator of the cell. As you can see, these chromosomes, they are arranging themselves in a single row at the center of a cell. So this alignment of chromosomes at the equator ensures that each daughter cell will receive an identical set of chromosomes during the later stages of mitosis. We also have spindle fibers that attach to the centromere. We said that these spindle fibers are formed by centrioles, right? So this is where our centrioles are located, to the opposite poles of the cell, right? So here, these yellow lines represents our spindle fibers. So as you can see, these spindle fibers are attached at the centromere of the chromosome. Alright, now moving on to the next stage of mitosis is we have anaphase. So in anaphase, this is whereby a centromere splits into two. So what do we mean by centromere splitting into two? You remember that our chromosome is joined together by a centromere, right? So when it splits into two like this, we're gonna have this chromatid and this one separating from each other and move to the opposite poles. Ne? So these chromatids, they are also known as daughter chromosomes, or you can call them sister chromosomes. So the spindle fibers begin to elongate the cell. So what do we mean by that? We mean that as these spindle fibers that are attached to the centrioles, as they shorten and pull these sister chromatids to the opposite poles of the cell, there are certain spindle fibers that elongate the cell, and this process leads to the formation of cleavage furrow, right? Now let us move to the last stage of mitosis, which is telophase. So telophase, chromosomes arrive at the opposite poles of and begin to decondense. If you remember correctly, at prophase we said that our chromosomes condenses and become visible. Now at telophase, after they arrive at the opposite poles, they decondense. And then we have a nuclear envelope material which surround each set of chromosomes. If you remember correctly, at prophase we said that the nuclear envelope and the nucleolus, they disappear. But then at this stage, which is telophase, is that nuclear envelope starts showing again, and also in nucleolus, right? And then we have the mitotic spindle, which is also known as the network of fibers. It is going to break down. Then we have these spindle fibers which are still pushing the poles apart. And then, as you can see, this cleavage furrow, the cells begin to break. Now, I said that this is the last stage of mitosis, right? But we have a stage which is known as cytokinesis. I said that cytokinesis is the final stage of the cell cycle. Now, moving on to cytokinesis. So cytokinesis is the last stage of the cell cycle, right? So what happens is that the cytoplasm is divided into two newly forming daughter cells. Remember, we said that mitosis is the process whereby a single cell divides, forming two daughter cells which are identical, right? So if you remember, this was a single cell, and then it started pinching in, forming two daughter cells. So, cytokinesis occurs when a cleavage furrow pinches in at the center of the cell, eventually dividing it into two separate daughter cells. So, in which stage? In anaphase, cleavage furrow formed. Now, at this stage, which is cytokinesis, it is whereby it pinches in at the center of the cell and eventually dividing it, forming two separate daughter cells. Now we have two genetically identical daughter cells, and these identical daughter cells have its own nucleus and a full set of organelles. If you can take a look, here we have three chromosomes which are red, and then on this side too we have three chromatids which are red, and also here we have three purple chromatids, and on this other set of nucleus we have three chromatids which are purple, meaning that the number of chromatids found in this cell is equal to the number of chromatids that are found in this cell. And if you take a look, I don't know, but our first, the original cell, our mother cell, also had these number of chromatids at the beginning of mitosis. So this is the end of our process called mitosis.
Sorry.
Now,
Sorry. Please go back to
okay, just leave your stage. Bye.
sorry. When I say cytokinesis falls under the phase which is telophase.
If I remember correctly, I said that cytokinesis right here is the last stage of the cell cycle, not mitosis. These are the stages of mitosis: prophase, metaphase, anaphase, and telophase, right? And then this stage is the last stage of the cell cycle. So a cell cycle has about 3 stages, right? Which is the interphase, the cell division, which is mitosis, and the last stage, which is cytokinesis. So as you can see, telophase is the last stage of mitosis, and this one, cytokinesis, is the last stage of the cell cycle. So to answer your question, cytokinesis is not part of the telophase. I hope this answers your question. Now, are you satisfied?
Yes.
Okay. Now moving on to the purpose or importance of mitosis. So mitosis is important because it ensures that the organism grows and increases in size. So what we mean is that mitosis increases the number of cells in the body, which is crucial for growth. For example, let's take how you grow up from being a baby to an adult. So what happens is that as bones lengthen during childhood and adolescence, mitosis generates new bone cells. So that's how growth occurs. So I also have a question for you guys. So think about a time when you had a cut that eventually healed. So, like, what do you think happened during that healing process? I want you guys to discuss and then give me an answer. Are you guys done?
I think what happens is that the cells are multiplied to replace those damaged cells. For example, if you have the problem with liver,
Yeah.
those tissues are able to regenerate themselves.
Okay, that's correct. Thank you. Reference for her. Okay, another significance of mitosis is that it results in asexual reproduction in some organisms. It can be plants or animals, right? So what we mean is that mitosis allows organisms to produce genetically identical offsprings without the fusion of the gametes. So by gametes we mean the sperm and the eggs. So I think we are done for today. I want to ask you guys some questions. Did you understand the lesson?
Yes.
Are you sure?
Yes.
So what happens during anaphase?
Anaphase.
Yes, anaphase.
During anaphase, the spinal fibers pull the chromatids that were aligned in the equator into opposite poles.
Okay. Okay. So what is the difference between a chromatid and a chromosome? Yes, sir.
Oh, okay, chromosome, chromatids, is a single, is one of the sister chromatids. A chromosome is made up of two equal sister chromatids, which is joined together with centromere.
Okay, so a chromosome is made up of sister chromatids which are joined together by centromere.
Yes.
Okay. So what is the function of a centromere?
Centromere joins or connects together the sister chromatids.
Okay. 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 Last question. What is the function of spindle fibers?
And the spindle fibers, they pull the chromatids into opposite poles.
Thank you. So I have a take-home assignment for you. So it happens that when an uncontrolled mitotic cell division, it is said that it results in cancer, right? So I want you to discuss how this uncontrolled mitotic cell division lead to cancer and also state the causes of cancer. Yeah. So to sum this all up, we said that a mitosis is a process whereby a single cell divides and forms two identical daughter cells. A chromosome, a single cell is going to divide and form two daughter cells. These two daughter cells will have the same number of chromosomes as the original or mother cell. And then we said that we have four stages of mitosis. Please make a toast. Please make a toast. PMET. Please make a toast. So we have prophase, metaphase, anaphase, and telophase. Here at prophase, we said that our chromosomes condenses and become visible. And then in metaphase, it is whereby they align and arrange themselves at the center of the cell. And then we have anaphase, whereby our chromatids are pulled to the opposite poles of the cell, and telophase, which is the last stage of mitosis. And in telophase, it is whereby chromosomes arrive at the opposite poles and then the cell begins to break. Such we also have cytokinesis, which is the last or the final stage of the cell cycle, whereby the cleavage furrow pinches in, resulting in two identical daughter cells which have the same or equal number of chromosomes as that of a parent cell, original cell. So, and also that since we importance of mitosis, we said that mitosis is important because it allows our bodies to grow, to heal, and also to maintain healthy tissues, right?
Yes.
So I think we will end here for today. Since we have come to the end of our lesson, I want you to write a class activity on mitosis. Next. Okay, you're supposed to write this for 5 minutes, and our period is almost over. So I will mark your books at the staff room, and tomorrow we will do corrections.
Okay.
you are asked to match column A with column B. So take for example number 1, you can say number 1A if you can see that on column B the matching answer is A. So it's about 1A. Understood? And then on question 2, you are given diagrams. So what you're going to do is to study the diagrams and then you answer the questions that are asked below the diagram. Yeah. You can write now. No copying. If you have any questions, feel free to ask.
So, if I have done question 1.1, can I show what I have to do it what?
Or let me see. Please. Let me see.
Explanation on what is asked in question
2.2. On 2.2, you are given 3 diagrams, right? Representing different phases of mitosis. So you need to study each diagram, see which phase it is, and then answer the questions that follow.
Okay.
Am I clear?
Yes.
Or do you also want me to mark question 1?
This one is correct.
No, it's not. The first question is not A. It's incorrect, sorry. But we'll do corrections.
Okay.
Today if I have time, and then if my period is over, we'll do it tomorrow. is over. So if you're done with the classwork, you can hand over the books. I'll mark them. Please come fetch them at break time. Okay.